Site that is jigyasa-csir, and you can find a lot of games and other interactive things, informatics, infographics, simulations and videos hosted over there. So you can visit our site. And I'm happy to say that we have reached more than 3 lakh students in past few years. So this is a short story about Jigyasa, how we are doing, how we are reaching to the society. So with that, without taking much of your time, let me introduce today's speaker, uh, Dr. Venkata Ramana Vemaluri. Dr. Venkat is a senior scientist in CSIR-IMTECH Chandigarh. Dr. Venkat has done his PhD in biotechnology from Jawaharlal Nehru Technological University, Hyderabad. Kothari postdoc. Just a minute, I will be sharing his biodata for you all so that you can read. So, yes. So Dr. Venkat has done his Kothari postdoc from University of Hyderabad. He worked for National Center for Microbial Resources, Pune. He also served as guest scientist at University of Stuttgart, Germany. Dr. Venkat has contributed to science by discovering more than 30 novel bacterial species and environmental microbiology. Dr. Venkat has published more than 30 papers in peer-reviewed international journals. His research interests are exploration and exploitation of anoxygenic photosynthetic bacteria for photoactive molecules and bioremediation of environmental pollutants. So this is the sketch which I read for you. So before I hand over to Dr. Venkat, I request all my friends who are attending this lecture to please stay on mute and switch off your cameras so that you can enjoy the lecture. Also, I request you all to please hear the lecture very carefully because we'll be having a quiz session after this lecture. And the top scorers of that quiz will receive a very attractive gift that you can use in your home or in your school or in your community to check the milk adulteration, and that will work for like weeks and months. So I request you to please attend the lecture very carefully. With that, I am handing over the stage to Dr. Venkat. Over to you, Dr. Venkat, please. Dr. Venkat, before you start, there is one more important announcement for our attendees. Please do not put anything irrelevant in the chat box because we will be sharing link for the quiz in the chat repeated time during the talk so that you can use that link in browser. But that link will accept the answers only when the talk is over. OK, so with that, I request Dr. Venkat to please deliver his talk.
I hope it is visible for you. Is it visible?
Yes, Dr. Venkat, it's visible.
Yes.
Dr. Venkat, your voice is low. Can you come closer to the screen a bit? And
is it okay now?
yeah, better. So you need to be a bit louder while delivering your talk.
Sure. Hello, good afternoon, students and friends. Today, actually, as Chandrasekhar said, we are delivering some lectures in the, as a part of Jignasa program. As a part of that, today I'm going to give you a glimpse of microbes. So the title is Understanding the Microbes. So the key objectives of this session is the lecture, followed by virtual demonstration of using milk adulteration kit, and discussion followed by the discussion, question and answer session, and then online quiz will be there on the topic which I deliver now, and later, based on the answers received, we will distribute the DIY kits to the quiz winners by post. So most of us know the importance of microbes. So we have to understand actually what about the microbes before we go into the details. So I'll give you a short idea, brief idea about the microorganisms. So microorganisms are a small creatures or living beings, so which cannot be seen with the naked eye. Okay, so they may be a single cell or colony of cells. And the microbes, the first time they have been observed by Anthony van Leeuwenhoek with the microscope in 1670s, and then followed by that, Louis Pasteur identified that, found that the microorganisms which are spoiling the food. But previously, there was a notion that there was a spontaneous generation, but by debunking that, the Louis Pasteur told that it is the food spoiling because of the microorganisms. And after that, 1880s, Coase discovered that the microorganisms also causes several diseases such as tuberculosis, cholera, diphtheria, and leishmaniasis. So all these microorganisms, actually when we look into the domain of life, there are the bacteria, archaea, and eukaryote. So as I show you, as you can see here, the bacteria and archaea are completely consists of microorganisms. But the eukarya, there are part of microorganisms and also macroorganisms. So such as fungus, yeast, protists, all this comes under eukaryota, which comes under the category of microorganisms. The rest of them are macroorganisms. So when you observe, actually, I just wanted you to know how these microbes look. When you observe in different magnification in different microscopes, different kinds of microscopes. You see, the first one is the phase contrast microscope. When you observe this microorganism or bacteria, a rod-shaped bacteria, the same bacteria under a phase contrast microscope, you will see the way which is shown here, which is a magnification of 10 to 100x. So when the same slide or same bacteria is viewed under transmission electron microscope, which is having a 1 to 2 million magnification, 1 to 2 million times of magnification, it appears like this. The internal organelles or internal parts, vesicles, DNA will also be visible in this case. And the other one is scanning electron microscope, where the magnification would be up to around 5 lakh times. And it doesn't actually, the scanning electron microscope scope gives you the surface view of the microorganisms in depth on the three-dimensional structures. So because I was showing these microorganisms, pictures, pictures in these uh, uh, microscopes, images, because some children are under the impression that the microbes, how the microbes look like. And because you see in the advertisements and all the cartoons of microbes will be shown in the type of cartoons. So going to details, actually the major, I just wanted to give you a brief idea about prokaryotic and eukaryotic cells. So I think you might have already, students might have already read in your books also, but still I just wanted to remind you, see what is the basic similarities and dissimilarities. The prokaryotic cells is different from the eukaryotic cell by not having a membrane-bound organelles and membrane-bound nucleus. It doesn't have, it's a it's a premier before nucleus. So these are more primitive than the eukaryotic cells. And this doesn't have membrane-bound organelles. And the similarities between these two is the plasma membrane, cytoplasm, and genetic material or similarities. But the lysosomes, Golgi complex, mitochondria, so these nucleolus, so these things are not similar among these two. Microorganisms, friends or enemies. So there may be miss. conception or notion that all these microbes are actually germs. Actually, because the way people uh, demonstrate in social medias and all, the germs are all bad. So all germs or all microbes are not bad. I'll give you some examples and show you how they are not bad and how they are bad. So they are both there. I mean, the microorganisms are friends as well as enemies. So how they are friends? You know, antibiotics. So what are the antibiotics? Antibiotics are the molecules or the compounds which are being produced by the microorganisms and also works against the microorganisms. So works against the pathogenic microorganisms. So it is actually in this case, these microorganisms are helping us in synthesizing or producing antibiotics. So similarly, these microorganisms that are present in the soil and the root nodules helps the plants in attaining the nutrients because these microorganisms what they do they break down the complex molecules complex nutrients into simpler nutrients which can be absorbed or taken by the plants. In turn, the plants also plants and the roots also give and support the host to these microorganisms in the soil. So there is a mutualistic association between the microorganisms and the plants in the soil. So in addition to these bacteria also useful. I mean, recently there were some reports that there are several bacteria that also degrade the plastic. So microorganisms also helpful in bioremediation. That is the remediation or degradation of toxic pollutants from the environment. And also coming to the other benefit, important benefits, they also act as a probiotics. So whenever anyone affected with the diarrhea, what the doctor actually prescribes the probiotics. What are the probiotics? So these are the good bacteria which are present in the gut of a human being. So these bacteria, when during diarrhea, these bacteria are washed off. So to replace those good bacteria in the gut, the probiotics is being given. So this is one of the important significance of the microorganisms. I give you a brief meeting about the gut microbiome in the coming slide. And coming to this, you see these microorganisms are used in food processing. In several food processing like soya sauce, making of soya sauce, cheese, yogurt, beer, fermentation, wine fermentation, bread, and etc. And also curd, yogurt, curd. So as I already told you, all these bacteria are not all these bacteria are not friends. There are also enemies, such as several bacteria, fungus, yeast. They cause several lethal diseases, dangerous diseases to the human beings as well as to the plants and also plant products and foods. You see here the lungs which are affected by the Mycobacterium tuberculate. So actually this is the human pathogen which affects the lungs and causes TB. And these are the Alternaria, Fusarium, Penicillium. These are the three different fungus which actually affects different food products. And you see the other bacilliform viruses, which actually affects the other plant products, like sugarcane also affected by some of the red rot diseases. So rhizopus is one of the common mold, green mold, which you observe on the bread. When you keep the bread for longer periods, then you will observe this green color molds on the bread, which is the which is because of rhizopus fungus. So similarly, there are several skin disorders which are caused by the yeast, which is the name is Candida. So similarly, there are several other illnesses are being caused by these microorganisms, pathogenic microorganisms. So, so what we have to do, we have to be careful about the avoid. We have to be careful and keep avoiding the pathogenic organisms and keep in touch and have the non-pathogenic and friendly organisms and friendly microbes. Why? Because you see now, this is a human microbiome. What is a microbiome? It's the total content of microorganisms present. It's called a microbiome. So how many microorganisms are present in a human? And typically, an adult human being has generate 10 trillion human cells. He himself has a 10 trillion human cells. However, the human microbiome in and around the human is around 100 trillion bacterial cells. So the bacterial cells present in and around the human body are 10 times more than the human cells. So what kind of organisms are present in this human body? The bacteria, archaea, fungi, protists, viruses. So there are several types of these microorganisms habitat in and around the body of the human body. So what is the relationship with this human? What are the relationships of these microbes with the human? Comments, either it is a commensal or mutualistic relationship or non-pathogenic and pathogenic. So commensals are the microorganisms, the relationship of microorganisms which they do not cause any harm to the host, but they get benefit from the host. So in the mutualistic relationship, what happens? They get the human body, humans get benefit from the microorganisms. In turn, the microbes also get the nutritive benefit and the habitat from the human body. So this is a mutualistic relationship where both of them are being benefited. I'll tell you how this is happening in case of human body. And there are non-pathogenic organisms which do not actually cause any harmful hazardous illnesses, but they attach to human body in one or the other way. And there are other pathogenic organisms also inhabit your human body, in and around the human body, which gradually being eliminated by your gut microbiome and other microbiota. This is actually, this also I'll cover in the next slides. So where these bacteria, microbes are present in the human body, where they are mostly concentrated. So they are actually habituated to, and they settle in specific parts of the human body, like skin. Some specific groups of microorganisms present in the skin. Some are mammary glands, some are placenta and saliva, oral mucosa, conjunctiva, and gastrointestinal tract. The way I'll tell you what are the groups of bacteria present in different parts of the body. So by looking at this picture, now you can tell me what this picture depicts. So you people can say a boy sitting alone. So is it real? Is it the true statement? No, because he's not sitting alone. He is sitting with his 100 trillion friends, of which are bacterial friends, are microorganisms which are present in and around his body. So nobody is alone until unless you have all microorganisms with you. Because if the man or the human being without microbiome cannot survive even for few hours also. So yeah, as I told you, different group of microorganisms are habituated to specific parts of the body. So we have a list of organisms which are actually abundant in particular specific sites of the human body. The list you won't be able to read all these things. Just to give an idea, I have pointed out here. What's the importance of human microbiome? So are these really invaders or beneficial colonizers? As I told you, they are not invaders, they are beneficial colonizers. So as I told you, these are microbiome is a set of organisms inhabiting in the human body. So the largest concentration of the microbiome present in the body, human body. So 90% of the microbiome present in the human gut. So rest of the microbiome, rest of the microorganisms are colonized in different parts of the body. So what they do, what is the major role of this microorganism? They are helping us in digesting our food and regulate our immune system. They fight with the bad bacteria that is present in our body, and also they help in digestion of some foods which cannot be digested by the enzymes that are present in the human body, and also protect against other bacteria that cause diseases. So they fight with the pathogenic bacteria and eliminate them. And also they mainly produce vitamins such as vitamin D, B12, thiamine, riboflavin, K, which are required for the metabolic activities of the body. When you read about amino acids, So there are different categories: essential amino acids and non-essential amino acids. So what do you mean by non-essential amino acids and essential? Essential amino acids has to be taken from outside or being synthesized by the microbes within the body. But non-essential amino acids can be taken even by the food also. So most of these microorganisms are helping us in giving the different vitamins also. So what happens is the microbiome same throughout your life? No, the microorganisms constantly evolve throughout your age and nutrition, your lifestyle, on hormonal changes and your heredity. So whenever, so your lifestyle decides, actually your good lifestyle decides what your lifestyle decides the your human microbiome, which in turn will be helpful for your health, health and sustainability. So always, and actually, in this microbiome, there are different types of groups of organisms. So a set of organisms, 90% of this kind of organisms will be helpful, and the 10% of these organisms in the gut or the body is helpful to maintain the balance. So when there is a dysbiosis of these microbiomes or microorganisms in your body, so it leads to the life-threatening illnesses. So always a balanced microbiota has shown to play an important role in health and sustenance. So this is the reason actually when there are several illnesses where people won't be able to won't be able to recover with the medicines. Then there are reports that they got recovered by the fecal transplantation. So what is the fecal transplantation? It's an advanced treatment of fecal transplantation, which is nothing but a microbiome of a healthy patient to the diseased patient. So you don't need to be embarrassed to listen these things, but it is the facts which is already going. The treatment is already going on in several countries. The fecal transplantation, the feces of a healthy donor, is being processed and then given as a capsules or an enema to the diseased patients. And there are so many reports where the patients have recovered unsuccessfully. Now we just go to the next part. So we have just till now we have an idea what are these microbes, what is the importance of these microbes, friends, enemies, and all. So how to identify them? So we also have to know how to identify these microbes. So before going to the identification, just I'll show you some pictures. If I show some pictures to you like this, you will and ask you what is this, you will just say this is a Mickey Mouse or Amir Khan and Eiffel Tower. It's a sunflower. So, and I'll ask you, I'll show you the one more picture and ask you what is this. And you may say it may be a bacterium, and you may say this is a fungus, and you may also say this is a yeast. So, if you say this is a bacteria, then I'll say this is a doll, and this is a tower, and this is a flower, rather than saying their names. So everything, every organism, the way every object and every man has a name, and each and every organism, also microorganism, also has their own names. So to identify that names, there are several. To identify those names, there are several methods to be processed. There are several methods. I'll show you one by one. Before I go to the identification, I'll just give you a glimpse of or idea about isolation of microbes. So generally, what the common basic principle of isolation is just taking a sample and putting it in saline or a sterile media and make it dilute into 10 times serial dilution and then plating on their respective media or agar media, lutein media. So you will find different microbes present in the soil will be grown on the plates with the serial dilution the way you have done earlier. So after doing this, you will notice different bacteria on these plates and the way I show you now. So, but you don't know the names of these bacteria. You may observe different colors, different morphologies, different shades. So how to identify? I just wrote here the names because just to have just to tell you what is this organism, but you don't know. Then what you generally go, what you people generally do, you have to go through a set of experiments in an orderly manner to identify these organisms. So how the first what generally people do? First of all, they do the gram staining, where I think gram staining is a common staining method and differentiating method for bacteria, whether it's a gram positive or gram negative. So it is well known that gram positive bacteria stain blue or purple because it has a thick polypeptide peptidoglycan layer and a low lipids. And gram negative bacteria stain pink, which do not have a very thin peptidoglycan and higher more lipids. And coming to the morphologies, the different bacteria. So after once you confirm this gram staining and you find out one gram stain positive and gram stain negative, and then looking at the under the microscope, you will see whether they are foci or rods or spirals. So whether they are a dual rod, dual sphericals or foci or multiple foci. So similarly, are they having multiple flagella, more flagella, or single flagella? So based on different morphological characteristics, we define these are the focus, these are the rods, these are the spirals. So similarly, if you go one by one, so the way you have you do here the morphological identification and then go to the physiological and biochemical identification. So all of these microorganisms have a specific role in in playing metabolic undergoing metabolic activities, such as several specific microorganisms produce specific enzymes, which itself can be differentiated from the one to one and the other organism. So identifying those enzyme production from that particular bacteria or yeast or fungus also helpful for identification and classification of these organisms into different groups. Similarly, there are several other patterns where some bacteria show this particular color in particular specific media, which we call as a differential media. So this differential media will differentiate the organisms based on their appearance on the media. So similarly, similar way to not only morphological, physiological, biochemical, and there are also xenotypic and phenotaxonomic. So there are all these characteristics cumulatively called as a polyphasic characterization. So till now we have discussed about this biochemical, physiological, morphological, and we'll discuss about the xenotypic in the future slides. If you keep on if you keep on if you keep on doing all the experiments, set of experiments one by one, so you see how many experiments one person has to do to identify an organism or a microorganism till the species level. So gram staining and then finding out the layer of this bacillus, which organism, is it positive for this enzyme? If it this may be organisms, and if it is negative for negative on these many organisms, again to confirm whether this is again to confirm these organisms to species level, is it a catalase positive or negative? So similarly, there are set of so many tests. I'll show you. Just go through quickly. So there are set flow chart. Identify the organism to the side. Are these really mandatory now to identify microorganisms? I say no, because now what happened to eliminate all these tests. So recent what happened, the techniques have been improved on identifying the organism based on the molecular methods. So you see here the advances in microbial methods since 1850, how they have developed. So starting culture dependent methods and microscopy, and then phylogeny, and then molecular methods. So I'll tell you how the molecular methods are easy and non-laborious and accurate in identifying the microorganism. So before going into that, we'll first just look at a DNA packaging and chromatin. So you see how the bacteria, when bacteria releases the DNA out of it, you see how much DNA is packed in the bacteria. So when this bacteria comes out of the cell, we see a huge DNA comes out of the cell then you see how the you will now shock. No, you will be shocked if look at this. How this much of DNA is packed into this bacteria. See this DNA is being packed into this bacteria is the supercoiling, because it is so. I mean the supercoiling is hundreds of times it is supercoiled within supercoiled DNA again supercoiled to form in loops and then compacted in the compactly packed in the bacteria. You see the DNA supercoiling is a second important way to compact the bacterial chromosome. So supercoiling within loops creates a more compacted DNA. So this is just a schematic illustration of how the bacteria, how the DNA is compactly packed in the cells, bacterial cells. So you see how some facts here. How long is your DNA? So if you see the bacteria, so what is the length of the genome? So the length of the genome is 1.3 kb to 14 MB, and it is 1500 to 7500 genes. So is 13.7 MB, the fungus around 36 MB, and Drosophila 12.1 MB, and human cell 1.5 GB. This is the size of the genome, that is a DNA. So you see the length of DNA in one human chromosome. If you see the length of the DNA in one human chromosome, it is equal to 2 inches, one chromosome. So the human cell contains 46 chromosomes. So one human cell contains 2.3 meters of the DNA. And one human being who is having a 10 trillion human cells himself, okay, is having 23 trillion meters of the DNA in his cells. So, which is equal to twice the diameter of the solar system. So, in such a way, the DNA has been compacted, the genetic material has been compacted in the cells. So now you'll come to the molecular identification. So there is a huge DNA. So how this molecular identification determines the bacteria, differentiates the bacteria or microorganisms. So you see, look at this fingerprints. So just to compare these fingerprints with the DNA, just assume that these fingerprints, the lines of these fingerprints are like a DNA. So are they look alike? Yes, all these fingerprints look alike to some extent, but there are some variations among these. So similarly, among the whole bacterial genome, there is a small part of the genome which is more, which differentiates the microorganisms of bacteria and yeast and fungus. So in case of bacteria, the small part of the DNA which codes for 16S ribosomal rRNA is actually the important molecular marker for the identification and differentiation of microbes. So Carl Hoos actually made the choice by analyzing this 16S rRNA in the bacteria and 18S rRNA in eukaryotes, and he actually classified the organisms based on these genes, these molecular markers, and differentiated and classified into groups. These actually 16S rRNA and 18S rRNA are present. So they are present in the ribosomes. You know the ribosomes are the protein factories within the cell. So ribosomes are present both in the both prokaryotic or prokaryotes and eukaryotes also. So among the whole genome, only 16S 1.5 kb, that is 1500 nucleotides length of the DNA actually determines or identifies which organism it belongs to. Okay, similarly 1200 to 1400 18S rRNA gene identifies to which eukaryote this organism belongs to. So what we generally do in the molecular identification, we amplify this particular gene. We amplify this particular gene out of the whole genome and we sequence it. Sequencing means, so we identify what nucleotides are present in that small stretch of the DNA, and we compare those nucleotides in the database, online databases, and the database itself tells this your sequence matches with so and so organisms, and it belongs to this particular group of bacteria or yeast or fungus. So when you do sequencing, you will get what is the DNA. DNA में क्या होता है? ATGC. So adenine, guanine, cytosine, thymine. So all these nucleotide sequence will get, and this nucleotide sequences when you what we do, we blast the nucleotide sequences in the online databases, and it itself show it belongs to so and so organisms, and to what extent it is similar, whether it is similar to 100% or 95% or 98%. So after you identify that the similarity, if you have a 3-4 organisms, we have isolated and want to identify the phylogenetic relationship, evolutionary relationship among all these, then what we do, the way there is a family tree, so similarly there is a phylogenetic tree which determines the evolutionary relationships between the microorganisms. So this phylogenetic tree is almost similar to family tree, but this phylogenetic tree based on the 16 SRRMA gene sequences which we identified. So this is also called as a dendrogram or phylogenetic tree. So coming to the methods used for removal of microorganisms. Okay, so I'll just give you a brief idea about how microorganisms can be removed or eliminated. So there are different methods for different purposes, because the food items are being sterilized in a different way. The microbiological media in the laboratory is being sterilized in a different way. The milk is being sterilized in a different way, and the glassware is being sterilized in a different way. So you see, this is, for example, this is moist heat sterilization, which is almost equal to a pressure cooker, but it has a pressure gauge also, which maintains 121 degrees, which attains 121 degrees at a for 15 minutes. So how it attains 121 under the pressure, 15 pascals. So this is one kind of a sterilization where all the microorganisms are eliminated, and the other one is the dry heat sterilization, where most of the organisms die, because we can glassware can be glassware which is used in the utensils, or glassware which are used in the laboratories can be sterilized by dry heat sterilization, which reaches up to 100, 200, 300 also. And the pasteurization is a process of sterilization in the milk. So milk is being treated. Actually, milk cannot be autoclaved or sterilized to 121 degrees. If anyone do that, then what happens? The proteins present in the milk also gets degraded. So what happens? Every particle of the milk is being sterilized. Subjected to 75 degrees for 15 seconds, which is called as a pasteurization, where most of the lethal human pathogens being killed at this particular temperature. And I think some of the utensils or some of the medical instruments, mostly syringes and medical utensils, are being sterilized with the UV radiation. UV also kills the microorganisms. So how these microbes are being preserved? We know the importance of microbes. So we also have to preserve the microbes. So how these microbes are being preserved for longer periods to use them in future. So actually the microorganisms can be preserved from short period to long period. So these are the simple slants, media agar, where microorganisms can be sprayed on to this media and put it in the 4 degrees only with weeks, or 1 to 2 weeks or 2 to 4 weeks. And after that, you will have to again subculture it again and preserve it. And when the other method of preservation is cryopreservation, where the microorganisms, bacteria or bacteria, viruses, yeast, fungus, can be suspended in the glycerol and stored at a -70 or -80, where it can be stayed active for more than 10 to 15 years. And the ultimate method of preservation is at liquid nitrogen, which attains a temperature of -196. And the other way of preservation is freeze drying or lyophilization. So in this lyophilization, what happened? Microorganisms are being treated with a skim milk and then dried it, under the vacuum and made into powder and stored at 4 degrees centigrade. So all these methods are very effective than storing it at 4 degrees centigrade. So coming to the other part of our session. So first of all, we also know what the adulteration, actually food adulteration is a major concern these days. Wherever you see, there is a lot of adulteration in foods. Either is in the case of poultry, either in case of vegetables or pulses or rice, anything. Adding adulteration is nothing but adding anything to the food substances or any substance to make it poor quality for their benefit, for marketing benefit, is called an adulteration. For instance, you see adding a certain natural and chemical dyes to attract consumers, and mixing of decomposed fruits and vegetables with the good ones, and mixing clay, pebbles, stone, sand, marble chips, and the other crop, and adding, and one more important thing is milk adulteration, oil adulteration. So many food products are being adulterated. So why actually are this being, why these products are being adulterated? So because practice is a part of business strategy, and also get the profits with the low investments and imitating the other foods, and also to increase the quantity of food products and sales. So we should be more aware of all this kind of adulteration. At least in this session, we are going to demonstrate one virtual. We are going to demonstrate you how to find out how to test the adulteration of milk. So after the presentation, I'll play a video where we have done demonstration as an experiment, and I mean you can also do that experiment at your end after you get after you receive kits. I mean the winners who receive the kits. So coming to milk adulteration, you know milk is considered as the ideal, one of the important and ideal food. Okay, so this is a fat, carbohydrate, protein content is more, and vitamins and minerals. So unfortunately, what is being happened? It is very milk is being adulterated throughout the world. So we cannot blame one person for adulterating the milk, because there are several middlemen. So farm workers, middlemen, companies are being, I mean, themselves are doing such kind of adulterations. So this leads to highly toxic, I mean, a toxic leads the adulteration leading to the toxicity of this food, leading to the several health issues, including nutritional deficiency, kidney disease, kidney disorders, and failures of individual organ systems also. What are the, I just give you an idea of what are the common adulterants used in the milk and their harmful effects. You see here, there are a list of some adulterants: alzarin, formalin, urea, starch, neutralizer, detergent, sodium chloride, skim milk, sugar and glucose, hydrogen peroxide. You really will be shocked to see such strong toxic compounds are being added to your foods. So what is the purpose? So one, because if the alzarin is being added, it is to increase the heat stability of the milk. So it can sustain the heat stability. It will not spoil even though in the higher temperatures. Okay, to maintain that they are using the alzarin and formalin as a preservative to make the milk. I mean, not to damage for. I mean to protect the milk from for more than 3 days or 2 days. They're using preservatives. The formalin is the better preservative which they are using, and it is very dangerous. And urea to increase the fat content, and starch increase thickness of the milk, the density of the milk, and also using neutralizers to regulate the acidity of the milk, and detergents to prevent curdling and increase the shelf life of the milk. And sodium chloride increase the density of milk, and skim milk increase the thickness and taste. So sugar and glucose is being used added in the milk to increase the carbohydrate content. So hydrogen peroxide to increase the shelf life. So all these things actually make them earn more and increase their business strategies and business, but spoiling the lives. So what are the side effects of these adulterants? We see all these three adulterants leads to damage of the intestinal tract and digestive systems, and these adulterants cause diarrhea, and these adulterants cause heart disease, cancer, poor eyesight, kidney issue, and sodium chloride, kidney diseases and headaches, and skim milk and sugars leads to the glycemic index, increase the glycemic index of lead, which leads to diabetes. And hydrogen peroxide, it's so dangerous because gastritis and inflammation of intestine. So what we have to do now? So we'll have to at least start with testing the milk, testing the adulteration of milk at your end, so that we'll start with this, and in future sessions we will also do some other experiments, demonstrations, and send some other kits also to do experiments at your end. In this session we'll do this milk adulteration test kit. So I'll give you an idea about this milk adulteration test kit, how it works. So the milk adulteration kit testing is a kit is designed for detection of 11 common milk adulterants in the milk, which I have already shown you the list. The principle, what is the principle of this? This kit is only a qualitative detection. You know what is the qualitative and quantitative. So qualitative is to identify whether it is present or not. Quantitative is how much it is present. But here we are doing only qualitative type of test, which indicates that whether the adulterant is present or not. And how it is actually, what is the principle of this kit? The adulterant plus if there is an adulterant in the milk, then that adulterant reacts with the given reagent and gives some specific color, and you will find out whether it is there or not. And that color you compare with the chart given over here, and you will find out whether it is adulterated or not. So there are different colors observed. There are different colors to identify the presence or absence of the adulterants. So there is a normal milk, one control or uncontrolled milk experiment. I mean test tube also there in all these experiments where there is where there is no presence of adulterant, and the others are presence of adulterants. And in the kit, in the given kit, all these test tubes and reagents and instructions, everything will be, and the chart, the color chart will be given for you, and with which you will be able to do at your end also, and send the results and encourage to send the results to us. And this is the instruction manual. You will find out in the adulteration kit who got it. And then you will be able to do the experiment very easily. So there are only few. So these are 11 different tests where there are where you need just heat, boil the milk, which after putting into the test tube. So this even can be done at your house in the kitchen also. Just put one bowl of water and boil it, and put your glass test tube in that, and for 5 minutes or 1 minute, wherever it is mentioned. The rest of the tests you need not heat or boil the test tubes. Coming to the other testing kits also, I just give you one more idea also. So there are coliform bacteria which are present in digestive tract of animals and plants, soil, and humans, and their wastes are also very much dangerous, and also the indicators of indicating organisms for testing drinking water quality. So whenever someone has to test a drinking water, whether it is potable or not, they test for coliforms. So we will do this coliform testing in the coming sessions. I'm just for an idea, I have shown these are the different coliforms. These are the total coliforms, group of large collection of different kinds of bacteria. Within that, there are fecal coliforms. And there are E. coli, which is the pathogenic organisms in this coliform. The detailed discussion about this will be done in the future sessions. So other basic experiments also you can do at your end, at your home with our help, provided with some media components and all from our side in future, such as effectiveness of different detergents or soaps in killing microbes. Whatever the soaps you are using or detergents you are using, are they really able to kill the microbes? And exposing bacterial plates to air, water, food. You can also expose the media to air, water you use, and test whether the organisms are there, or bacteria or yeast or fungus growing or not. And effectiveness of floor cleaners, and effect of temperature on microbial growth. So contamination, food spoilage. So at what temperature, when you are putting your food in the fridge, so whether how many days it is taking, if it is not putting in the fridge, what is the temperature of the fridge? So why it is spoiling? And also you can do other experiment, other one more experiment like sanitization, effect of sanitization in your hands. So you can check your microbial growth in your hands before sanitization and also after sanitization, after washing and before washing. So all such experiments will be covered in the coming sessions. So I just wanted to give you a brief idea about microbial resource centers. So as I told you, this bacteria or any bacteria which is useful during our research or during exploration or experiments, if you find out any bacteria, so many people isolate bacteria. So where these bacteria are stored? Are they being stored at their laboratories for longer periods? No, they don't have. All small laboratories may not, schools and colleges may not have a sophisticated equipment to maintain them for the longer periods. So what they do, they send to a microbial resource centers like microbial type culture collection and gene bank, which is called as MTCC, from where now I am talking to you. So I am also working in an MTCC, which is called as a microbial type culture collection and gene bank. So what is it meant for? It is a microbial resource center where we preserve and keep, maintain all these microorganisms received from different people and also supply these microorganisms to anybody who wants to do research or anybody who wants to test, who wants to do experiments to colleges, schools, industries, everywhere. This is the mission and vision, and this is the microbial type culture collection in the Chandigarh, which comes under, which is the under the umbrella of CSIR-IMTECH Institute. CSIR-IMTECH is the Institute of Microbial Technology. And in this MTCC, we deal with, we have yeast, fungus, bacteria, actinobacteria, anaerobic bacteria, photosynthetic bacteria. So different kinds of microorganisms we preserve here. So we also do services. If anybody isolated bacteria and they don't know how to identify them, they can send us and ask us and take the services from us to identify them. So we will charge we will charge nominal prices for the academic organizations, and we'll do everything. We'll do the characterization or identification from them for them, and we'll give all the details to the any academia. And we have such a sophisticated equipments over here. So where we will be able to identify 96 bacteria in a 1 hour. So by a Malditov biotyper, it's an instrument where we can do that. There are also other equipments where we will be able to do 48, 25 bacteria in a short span of 8 hours to 6 hours or 24 hours. This is the lyophilization facility in the MTCC. You see, these are the vials stored as a freeze-dried forms. So lyophilization also called as freeze drying. So in MTCC, there are 12,000 stakeholders across the country are dependent on MTCC for their academic and research. In addition to this, in addition to supplying the bacteria or microorganisms and preserving the microorganisms, the scientific community in the CSIR-IMTECH and MTCC are explore novel microorganisms for their potential applications. So till now, more than 150 to 200 novel microorganisms have been explored in this institute. So I end with this. I end my talk with this slide, and it's open for any questions. So after this, one thing I'll do. First of all, shall I play the Shaker? Shall I play that video?
Yes, yes.
Yeah. Is it audible? I mean, is it visible?
Not as of now.
First of all, I have to. I think just wait. I'll have to close.
Stop sharing. You have to stop sharing this PPT.
Yes. I think now it's working.
Today we will perform the milk adulteration testing to check the presence of adulterants such as urea, detergent, starch, sodium chloride, etc. in the milk. For this we will use high media milk adulteration testing
Is it
kit,
audible?
which contains all
Yes.
the reagents required for testing of 11 adulterants that are added on regular basis by the middlemen and unfaithful farm workers. As you can see, the kit contains the reagent bottles, measuring test tubes, measuring test tubes, measuring cylinder, gloves, and the procedure and the result interpretation chart. First, we put on the hand gloves. Then we need to prick these reagent bottles using this pricking needle which is provided in the kit. Then arrange and label the test tube according to this colored chart which is provided in the kit. So first we will start test the presence of alzarine in the milk. Take 1 ml of milk in each test tube using this dropper which is provided in the kit. Then add 1 ml of alzarine reagent in the test tubes. As you can see in this color chart, the normal milk change its color to red while the alkaline milk change its color to magenta. As you can see in this test tubes, the normal milk turns red while forms a red color ring, while the alkaline milk forms the magenta color ring, which marks the presence of alzarine in the milk. Now we will test the presence of formalin in the milk. So, as mentioned in the procedure, take 2 ml of milk in a test tube. Now add 2 drops of reagent FM1 in both the test tube. Mix well. Then add 1 ml of reagent FM2 from the side of the tube, slowly. Then observe the color. Ring formed at the junction. As you can see in this color chart that for the presence and the absence of the formalin, the rings are formed at the junction. But here, instead of forming at the top, it's formed at the bottom. So as you can see that the ring is formed is little bit of reddish or purplish in color, which marks the presence of formalin in the milk. We will test the presence of urea in the milk. So we'll take 2 ml of milk in the test tube. Then add 1 ml of reagent Q in the test tubes. As you can see in this color chart, the presence of urea is marked with the presence of yellow color and the absence is marked with the white color. Now we will test the presence of starch in the milk. So take 2 ml of milk in the test tube. And the second step is to boil it. So either you can use the water bath or you can boil the water and then keep it in the beaker and then dip this test tube in that water. After boiling for 5 minutes, Now we have to add 5 drops of SG reagent which is provided in the kit. So you can take out this reagent and measuring cylinder. Now pour very carefully. As you can see in this color chart that if the starch is present, the milk will turn to blue. So as you can see here that there is no such coloration which marks the absence of starch in the milk. Now we will test the presence of neutralizer. So we will take 2 ml of milk in a test tube. Then we will add 1 ml of N1 reagent. Then add 3 drops of reagent N2. As you can see in this color chart that in the presence of neutralizer the milk will turn pink and in the absent it will turn orange. So as you can see here that from the pink color you can say that the milk contains the neutralizer. Now we will test the presence of detergent in the milk. Take 2 ml of milk in each test tube. Then add reagent DET, 2 drops of reagent. As you can see in this color chart that in the presence of detergent the milk will turn into purple color. So here in the middle one we show the purple color which marks the presence of detergent in the milk. Now we will test the presence of sodium chloride in the milk. So for this we will take 2 ml of milk in the test tube. Add 1 ml of reagent NCl1. Then 3 drops of NCl2. As you can see, in the presence of sodium chloride, the milk will turn yellow and the absent it will turn brick red. So here, as you can see, the presence of yellow color marks the presence of sodium chloride in the milk. Now we will test the presence of skim milk powder in the milk. So we will take 2 ml of milk in the test tube. Now we will add 1 ml of reagent SKN1 in each test tube and 1 ml of SKN2. Mix the content thoroughly and keep its test tubes in the boiling water for 15 minutes. After cooling of test tube, you have to observe the change of color. So as you can see that in the presence of skim milk powder, the milk will turn the blue and in the absent it will turn green. As you can see here that the milk will turn into blue color which marks the presence of skim milk powder in the milk. Presence of sugar, that is sucrose, in the milk. So we'll take 2 ml of milk in the test tube. Then add 1 ml of reagent SGR1 in both the test tube and 1 spoon of reagent SGR2. Now boil the milk for 1 minute. So as you can see, in the presence of sugar, the milk will turn into red or brownish color. So as you can see here, that the brownish color marks the presence of sucrose in the milk which is used as an adulterant. Now we test the presence of glucose in the milk. So take 1 ml of milk in the test tube. Then add 1 ml of reagent GL1 ml of GL2. Then boil these test tubes for 5 minutes. As mentioned in the colored chart, that in the presence of glucose, the milk will turn into blue, and in the absent, it will turn into greenish. So you can see here that the bluish color marks the presence of glucose in the milk. Now we check the presence of hydrogen peroxide in the milk. So we take 2 ml of milk in the test tube. Then add 1 small spoonful of reagent HP. It's powdery in nature. Okay. Now wait for 5 minutes and then observe the color change. As you can see in this color chart, that if the hydrogen peroxide present, the milk will change its color to blackish. So here, after testing, the milk changed its color to black, which marks the presence of hydrogen peroxide in the milk.
Yeah, thank you, Dr. Venkat.
Yeah. One second.
Yeah.
Thank you. To clear everything, one doubt to most of the students, which most of the students might have. See, most of the students might be thinking that what are the milk we tested? So there are 11 adulterants we found. So don't, misinterpret that the milk we have tested has a 11 adulterants. Actually, for the sake of understanding, we have taken one set of milk and added all 11 adulterants from the lab and showed you the comparison between the positive and negative. Okay, so in your case, you need not do both. You need not do both the control and positive control. You can directly you can directly take the milk and add the respective reagent and do the experiment. Just find out. So just we added only to for the sake of understanding and show the visible difference for you students. Yes, Shekhar.
Yeah. Okay. Thank you, Dr. Venkat, for the informative talk and the demonstration as well. It was a learning experience for me as well. With that, I would like to keep the, make this floor open for the questions. So anyone have the question can ask in chat or otherwise raise their hands. I'm lowering all the hands for once, and then if someone has the question can raise their hand there again. Just give me 1 minute, so I'll be lowering all the hands. So if anybody have some question, can raise a hand, and I'll be happy to give the mic. So I believe Amit Kumar have a question. Yeah, Amit, please unmute yourself and ask your question. And if Amit is not around, Rohan Kumar Singh. Rohan Kumar Singh, if you have any question, you can ask. If Rohan is also not around, Anupam Kumar. Is Anupam around? So there are a few questions in the chat. People are asking, can we do these experiments at home? Or where can we get these testing kits?
It is actually clearly mentioned to them. I mean that we will have a, we'll send a link for the quiz, and the quiz winners will get testing kits to their home. Will be sent to their home.
Okay. Okay, I believe I can add to your question that yes, these are we bought these kits from these kits, customized kits from a particular vendor. You can also search on the net and buy these kits if you don't win in the competition. So if there are no more questions. Please put your question in the chat because there are many raised hands and people are not around.
Somebody was asking how microbes help in degrading plastics.
Yeah.
Yeah, microbes help in degrading plastics. I mean, there are only few reports that the enzymes responsible for the degradation of plastics are being present in those microorganisms, which break down the complex plastics into the simple.
So those who raise their hands, I allowed the mic to all those people who have raised their hand. So if anybody is around, can ask the question like Rohan Kumar, VK729117, Chikla, Devika, P. Tulsi Ramani, Ujjwala Ray, Dimahi Brahmbhat.
Dr. Venkat,
Yes,
there is another question on the chatbot which says, are there only 11 adulterants for milk?
no, there are several other adulterants also. The kit which we have demonstrated having 11. Other kits also have more than 11, 20 adulterants. It depends on the, the more the testing adulterants, the more the cost.
Sir, I have a question.
Okay, yeah, please go ahead.
Okay.
Sir, why adulteration are harmful for us?
So how? Because these adulterants which are being used are toxic compounds, are the chemicals. They are not organic compounds. So these chemicals actually, for the sake of their increasing the shelf life and and increasing the heat stability and preservation for longer time, they are adding for their purpose. What happens? These chemicals, if you directly add some chemicals in your food and keep on eating every day, what happens? Your biological system will get deteriorated. You got my point?
Yes, sir.
Yeah.
Thank you for the answer.
You're welcome.
So, there are no
Sir,
more.
can I ask a question?
Yes, yes.
Yes. Those who have raised their hands, I allowed the mic to all. They can ask. Yes.
Will it harm us while adding chemicals, sir?
No, you need not. You need not touch any of these chemicals. Okay, all these chemicals comes in the reagent bottles. You just have to put it by wearing gloves.
How sodium chloride and sugar can be adulterants?
Of course, they are also identical. They are the sugars, na. So if you add more sugar to that, it increases the density and thickness and carbohydrate content rather than the protein and actual composition of the milk is being destructed. So you see, then the cheapest material, adding a cheaper compounds, food compound products in the milk, and getting benefited for, and they are getting benefited, and not we actually who is consuming.
Sir, can I
Thank you,
ask a
sir.
question?
Yes.
One by one, please go one by one. Yes.
Sir, how many human cells are there in a human body?
That is what I told you. 10 trillion human cells are there in the human body.
Sir,
Thank
can I
you,
ask a
sir.
question?
Yeah, please go ahead.
Sir, in one slide, I saw that are microbes good or bad?
Yes.
But some microbes are good, but some microbes are bad. So what is the definite answer of that? Is microbes good or bad?
Of course, all microbes are not good. All microbes are not bad.
Yes, sir.
So you see, there are bad people and also good people. So how can you define the human beings on the same platform?
Okay, sir. Thank you, sir.
Okay, I believe we can switch on our cameras for a group photograph. Let's have a graph
Shekhar,
paper.
may
Don't-
I ask one question to Dr.
Yes, yes, please.
No, because while I was seeing the experiment, the demonstration that was going on, I just had a question. Is there a minimum amount of these adulterants that should be present in the milk for this kit to identify them?
Yes, yes.
You know, what is the minimum concentration?
Since it is not a quantitative test, we approximately the detectable amounts like 1 gram, 1 ml per 2 ml of milk have been added to that. So in one case, what happened in case of starch, I think trying to add a control for a positive control, we add some other similar compound of starch. It was not worked out. So definitely detectable amounts to be needed. I mean, detectable in minor amount only. If it is a quantitative, then we would have quantified, but since it's a qualitative, we approximately add.
All right. Thank you.
So I have allowed mic to everyone who has raised their hand. Do you have any question? Please go ahead. Otherwise, we will have a group photograph and we will.
Sir,
Have,
is there,
yes.
sir, is there any advantage of adding adulteration?
Your question was not clear. Please ask again.
Sir, is there any advantages of adding adulteration? Unknown number.
Your
We are
voice is
losing your losing your voice in between. We couldn't hear you.
Sir, may I ask? Sir, is there any advantages of adding adulteration?
no advantage is not for us. Advantage is for vendor. Disadvantage is for the user or consumer. Okay.
Okay.
So I request you all please switch on your camera so that we can have a group photograph. Please all switch on your cameras and then we'll be moving ahead to the next section of this event. We'll have an online quiz. I'm giving like 30 seconds to you to switch on your camera so that we can have a good photograph.
Sir,
Yeah, I can see a very
is
very
the
less.
variant virus?
Yeah, please go ahead with your questions. Don't stop. Please go ahead.
Shekhar, there is one more question is, can we identify useful and harmful microbes by simple observation of microbial strains?
No, not at all. We cannot differentiate non-harmful and useful microbes based on the morphological observations. Definitely, there are certain set of tests which has to be carried out to identify whether they are harmful or beneficial.
Sir, are there any adulterations that are useful for us?
Sorry.
Sir, are there any adulterations that are useful for us?
No, adulterant is actually an additional substance which decreases the quality of the product. So why there will be no use of adulterants? We need a pure quality. Adding any adulterants decreases its quality. It's a mixing. In Hindi, what we call
Microbe is in our food chain, also in our environment.
yes, of course.
Thank you, sir, for the answer.
There is one more question which says which bacteria is responsible for setting off curd without adding curd to the milk?
Setting of
Which bacteria is responsible for setting off curd without adding curd to the milk?
that is what lactobacillus. Actually, lactic acid bacteria, lactobacillus, which is responsible for making the curd.
And there is another question which asking, does everyone has the same microbes?
Of course, definitely not. Definitely not. Not everyone has the same composition of microbial composition varies from person to person.
Okay, yeah, okay. I believe we need to stop our question round here because many students are waiting for the quiz session. So we are making the quiz live for you all. Please attempt the quiz wisely so that you can win the prizes.
You tell them the time given for the quiz to complete it.
Yes, so quiz will be remain open for next 20 minutes.
There are 15 questions.
So it's 4:26. So we will close it at 4:41. Sorry, 4:46.
And also ask them to mention their minimum details to identify them.
Okay. So that's very much required because please fill your details carefully because we need to send those prizes to your address. So we need your complete address, complete postal address. So fill carefully and all the best to you all.
Sir, my chat box is not opening.
How can I help you? Okay, so just you need the address for the quiz form.
Yes, sir.
Okay, type BIT. Is it done? BIT dot.
Yes, sir.
LY slash Jigyasa, capital J I G Y A S A hyphen I. I'm telling this is in capital I M T E C H. So I'm repeating it again in small B I T dot L Y slash capital J small I small g small y small a small s small a hyphen in caps IMTECH.
Sir, how can we go to attend quiz?
Just use this link in your Chrome, in your web browser, and attempt the quiz. I'm sharing the link in PPT also. You can see. Is it visible now?
Yes, sir.
Yeah.
Sir, we will click this link on the Chrome and attend the quiz.
Yes, yes. Please don't waste your time in discussing the thing because people are attempting the quiz and they are far ahead than you. Please do it.
Shekhar, somebody is asking which email ID we have to put.
Any working email will work, no problem. Any email which you use, yes, anything, any personal or official mail which works, because we will be sending you information that you have won. Yes, the quiz is only for students.
Yeah, I have replied.
Yeah. Yes, your school address, We want your principal, your teacher gifting you in front of your friends. So we want your school address. Don't put your home address. It's very categorically mentioned over there, school name and address.